We're going to be going over the physiology of the nervous system, specifically looking at how neurons work. And we want to look at a few ideas, such as the resting membrane potential, the graded potential, and also, of course, the action potential. In order to do this, we want to look at the different ions that we're going to use to do this. This is the electricity of the biological nervous system, and to do this biological electricity we need ions. And more specifically, the ions that we look at are going to be sodium, Na+, potassium, K+. We'll also see chloride, which is Cl-, and we'll also see calcium, which is Ca++. And in order for these ions to be able to cross in and out of the cell, to move into and out of the cell, we also need channels. And the channels that we're going to look at are going to be the ligand gated channels, also the voltage gated channels, and we may also see the mechanically gated channels and leak channels. So let's orient ourselves to this picture to see what we're looking at. This is a neuron, and this is known as the cell body of the neuron, where the nucleus is. And within the cell body, you have dendrites, which are these spiny looking things that receive the input from other neurons. And so the input is coming in through these dendrites. The cell body is going to get stimulated by this through some electrical activity and possibly send a signal of its own through this part of it, which is known as the axon. The first thing to remember about the transport of ions from one side of the membrane to the other is the sodium-potassium pump and remembering how it works. So the sodium-potassium pump, which in this image is depicted by this circular protein right here, embedded within the membrane of the neuron, takes 3 sodiums and pumps them out of the cell. It also takes 2 potassiums and pumps them into the cell. Now it does this using energy. So let's not forget that this is using ATP, and that ATP gets converted to ADP plus P. And that's where the energy is coming from, through that ATP hydrolysis reaction. Now, what's going to happen here is that because it's doing this, and you can see it happening all over the neuron here. So I've drawn it over here, I've also drawn it here, and you can imagine it all over the place. What's going to happen is that as it does this, it creates an environment wherein there is a lot of sodium that is going to be outside the cell in general. High sodium levels are found outside the cell, and this will be all over the place, right? Because let's not forget, these sodium potassium pumps are going to be everywhere. And simultaneously, a lot of potassium will be found inside the cell. And again, this will be all over the place within the cell. So what does this do for us? This creates a potential, a type of potential. And what that means is that, if you recall, small entities like ions like to be in equilibrium on both sides of a membrane. So that if you have a lot of sodium on the outside, it's going to want to go in. If you have a lot of potassium on the inside, it's going to want to go out. So by using this sodium-potassium pump and creating this situation where you are not in equilibrium, this will create a potential, meaning that now if we allow for the sodium to go in, it will go in. If we allow for the potassium to go out, it will go out. Let's look at some of the other channels that we have here, some of the other proteins within the membrane that are also at work here. So this pink looking protein right here is known as a potassium leak channel, whereas this purple one on this side is known as a sodium leak channel. And so here we have, for example, our channels. Let's recall we have ligand-gated channels, voltage-gated channels, mechanically gated channels, leak channels. So this will be the first one that we talk about at the bottom, this leak channel. And the difference between these two channels is that in general, the potassium channel is more leaky. And so what's going to happen is, as the sodium-potassium pump pumps the potassium in, some of it will tend to leak out through that channel and find itself on the outside. Still, most of it will still stay on the inside, but some of it will leak out. And the same will happen with sodium, but to a lesser degree. Some of the sodium will end up leaking in through these leak channels. We should say, however, that the potassium leak channels are much more leaky than the sodium channels. This will be important when we discuss the resting membrane potential, which is coming up very shortly. So for right now, just keep in mind that you have this active transport using ATP energy, pumping 3 sodiums out, 2 potassiums in, and then there's some leakiness going on around that. There's a potassium leak and there's a sodium leak. And the potassium leak is stronger than the sodium leak. What else can we say about this picture? Well, notice that there's also chloride here in green, which is negatively charged. And chloride is found more so on the outside of the neuron, but you also have some of it on the inside of the neuron as well. This is not going to contribute heavily to the resting membrane potential, but we include it in the overall picture because it does give some effect. What gives much more effect, however, are these proteins here. This is a protein right here, and this one is a protein as well. Now, what you notice is that I drew negative signs on those proteins. Those dashes are negative signs. So you'll have a protein, we can draw it like this in any sort of random way, and then we draw the negative signs on it because it shows that these are negatively charged proteins. Now, proteins are much, much, much bigger than ions. So whereas ions can leak through channels and move across the membrane, proteins cannot. So this protein is going to be intracellular, and that makes sense because after all, cells make protein. Let's not forget DNA, mRNA, protein, right? So cells are always building proteins, and proteins tend to have this negative charge on them, and then they're stuck within the cell and they cannot leave the cell. And they give this negative charge to the inside of the cell. The next thing we want to do is look at this device right here. This is a voltmeter. And what it does is that it has a sensor that goes into the cell, which is this pointy one right here. You can see it's going into the cell. And then it has another sensor that stays outside the cell right there. And these two sensors are going to see what the difference is between the inside versus the outside in terms of voltage. And this type of voltage is an electrical idea in terms of the potential that I was referring to with these charged particles either inside or outside of the cell. Now notice that this right here, this voltmeter that we use, and this is done through experimentation by scientists. They use these devices to see what the differences are inside and outside. And you can see that this voltmeter in millivolts, little m, big V right there, has a line going to minus 70 millivolts. There's a minus 70, there's a minus 30, there's a 0, and there's a plus 30. We're going to zoom into this so you can see the details a bit better in a second. But for right now, just realize that this right here, this voltmeter, is going to tell us what the voltage is on the inside of the cell with this sensor versus the outside. To do this, let's zoom in, and we're going to use this picture. Notice that we're taking this part of the membrane and we're zooming into it, and now we're looking at it in a larger way up here. And so you can see that this is the pink channel, this is that blue one, and this is the purple one. So we've got the sodium leak, we've got the sodium potassium pump, and we've got the sodium leak, and then we have that voltmeter, and here you can see its detail. Also, you can see how that membrane of the neuron is, of course, made up of phospholipids. Once you get close to it, you'll see that membranes are always made of phospholipids. So let's take a look at this voltmeter and try to understand what it's telling us. We've got a sensor that goes into the cell. You can see the tip of this pointy sensor going into the cell through the phospholipids, through that membrane, and this sensor is outside. What does this minus 70 mean right here? It means that the inside of the cell, right, this is the inside, and this is the outside. It means that the inside of the cell is negative compared to the outside. Let's try to understand what this means, this negative 70. Well, first of all, let's remember what the sodium potassium pump is doing. The sodium potassium pump is pumping out 3 sodiums and is pumping in 2 potassiums using ATP hydrolysis. So you want to think about it this way. First of all, if 3 positive things are going out every single time 2 positive things are going in, then that means that the outside of the cell will tend to be more positive than the inside. Another way of saying it is that the inside will tend to be more negative, which is what this is telling us. This is about what's going on the inside versus the outside. Let's not forget that. The inside will be more negative compared to the outside if the outside is more positive compared to the inside. So you can see how this sodium-potassium pump is starting to push us towards this negativity on the inside of the cell. Okay, so we know that we're tending to become more negative, and we call this polarization, right? When you get away from zero and you become more negative, we call it polarization, more polar towards the negative side. On top of it, we already mentioned that the potassium leak channels are more leaky more leaky than the sodium leak channels. So you have more leakiness going on with the potassium. That means that more positive stuff is leaking out compared to positive stuff leaking in. Well, if more positive stuff is leaking out, that means the outside becomes even more positive. Or, if you think about it differently, the inside becomes even more negative, right? So it keeps moving this way as these leaky channels have their effect as well. The other thing we mentioned was that we do have chloride, which tends to be more on the outside, and it is negatively charged, but because chloride doesn't have a strong effect, even though there's more of this negative on the outside, it doesn't really move the needle too much. So it doesn't have a big effect. What has a very big effect, on the other hand, are these negatively charged proteins that you see here that we referred to before. So these big things are proteins, right? And you can see that these are the negative charges on them. And remember that these proteins are too big to cross the membrane, so they're stuck on the inside. And these negative charges really are going to build up within these proteins, bringing that needle ever more towards this negative 70 value. Now, this negative 70 value is a value that you should memorize. This is a very important number to memorize, this negative 70 millivolts. This is known as the resting membrane potential, or RMP. Resting membrane potential is negative 70 for most neurons on average. If we plot these values on a graph, and we represent the voltmeter on a graph, we'll see something that looks like this. So notice this is the voltmeter, and it's telling us that our resting membrane potential, RMP, is at negative 70. So the idea is that is where neurons start before they become electrically excitable. So if I were to look at this graph right here, notice I've got these two axes. On the horizontal axis, I've got milliseconds. Milliseconds. M-sec. Milliseconds. And on the vertical axis, I've got millivolts. M-V. So let's not forget what milli means. Milli means 1,000th of a second. And millivolt means 1,000th of a volt. So we're talking about very small values, both in terms of time and in terms of voltage. But these small values are enough to allow for the biological electricity to do what it needs to do. And so what you want to think about is that you begin on this resting membrane potential of negative 70. That's where you begin. So if you're a neuron, you are resting right here. And unless you become stimulated, you will stay on this negative 70. This is your resting membrane potential. You will just stay here unless another neuron tells you to do something. So notice this is resting membrane potential, and we also say that because we are negative, right, we are also polarized. We are polar, right? So this polarization, kind of, if you think about North Pole, South Pole, that positive and negative of a magnet, right? That's a word that polarized is used throughout science in terms of having this negativity on it. And so that's what we're using this word for, is that this negativity of the inside of the cell, meaning it's polarized. We also use the word potential for resting membrane potential because the idea is that we have a potential ability to depolarize. In other words, to go from this negative and to start moving towards zero, and that would be a depolarization. So if, and we're going to see this, if we see that the charge on the inside of the cell starts to move towards zero, right, moving up towards zero, then you are taking away that polarization, right? And so we call this, moving up, we call this depolarization, right? Depolarization as you move towards zero. What we're going to see is that this happens so fast that sometimes we actually go past zero, all the way to that plus 30, which we have up here, right? And then from there, this overshoot, it's still called depolarization just because it's still part of that upward movement. So just keep that in mind. Don't get confused by that. Depolarization is taking away that charge, but it just overshoots. Things happen too fast for it to just stop at zero, basically. So anything that goes up, we call it depolarization. And then in order to drop back down to our resting membrane potential, because we want to be able to do this again, right? Then we drop back down. And what we'll see is an overshoot also on the way back down, where you kind of go past this negative 70, right? And anything moving downwards back to that resting membrane, we call that repolarization, right? We call it repolarization. And so you can see that basically the idea is depolarization and repolarization, going back to eventually our resting membrane potential, because that way we can do it again, right? And then you can imagine it depolarizing and repolarizing again. So keep this in mind. We're going to understand this better and better as we look at more and more details about how the neuron works. Just keep these ideas in mind for now, and then the details will come in a little bit. So just to recap everything, we've got this neuron that has sodium-potassium pumps everywhere, has potassium leak channels, sodium leak channels, has chloride as well, which does not contribute too much to the resting membrane potential. It also has these negatively charged proteins, which contributes a great deal to the resting membrane potential. And then we've got this voltmeter, which tells us that we have a potential of this negative 70 millivolts on the inside of the cell that can now get depolarized to basically create some sort of signal in this cell. So we really want to understand this signal. What does it mean for this cell to depolarize? and then possibly give us a graded potential, which is a potential that stays more local within the soma or the cell body itself and does not propagate through the axon, or a more powerful version of it known as an action potential, where this cell body gets depolarized enough to where the signal does propagate down the axon and actually will stimulate another neuron further down. So that's what we want to figure out next, is graded potentials and action potentials. And we want to keep all of this in mind as we move forward. So the next thing we want to see is taking away some of those details that we drew in, but keeping some important things still there, and also adding a few things to our picture. So notice what I did here. I removed some of the clutter that we saw before, but I also ended up adding this neuron down here to the picture. So we can call this a presynaptic neuron. What is a presynaptic neuron? Very simply, it is the neuron that will stimulate the next neuron. Right? So if you think about it this way, you've got a presynaptic neuron, then you have the synapse, which is the space between one neuron and the next, and then you have the next neuron in line, which you can call a postsynaptic neuron. Right? So this one now, we can call it a postsynaptic neuron. Right? So the idea is presynaptic neuron, synapse, postsynaptic neuron. If we look at the presynaptic neuron, we notice that it, of course, also has a soma, or sometimes called a cell body. And so the same details that you see up here with these channels, the sodium potassium pumps, the channels, the proteins, all of that will be in here as well. But we're not going to draw it again, but you just apply the same thing here. Now, you want to imagine that this presynaptic neuron is able to get an action potential, which we're going to describe very shortly. And an action potential is, for now, just think about it as a signal that moves down the axon. And so you can see how it's going to move down this axon and keep going and keep going. One piece of axon at a time, basically, until this axon branches into axon terminals. All right, so these branches are called the axon terminals, and then these axon terminals will allow for the signal to move down those axon terminals. All right, so you've got the axon terminal, and then over here you have this swelling there known as, I'll write it over here, as the synaptic end bulb, right? This part right here is the synaptic end bulb. I wrote it over here just in order not to clutter the picture. But that's what this swelling is here. And then these red dots are the neurotransmitter, which you are already familiar with if you did the muscle chapter, right? This neurotransmitter here, you learned it as acetylcholine. And acetylcholine, sometimes we just say ACH, right, for acetylcholine. We saw it with the muscle chapter, and we can think about it again in the nervous system chapter because neurons use it as well, just as well, to communicate with other neurons. But also remember for now, for this chapter, that ACH is just one of many neurotransmitters for the nervous system, right? So you've probably heard of dopamine, serotonin, norepinephrine, and the list goes on. So there's many different flavors of neurotransmitter that we can use here for the nervous system. But if acetylcholine is comfortable for you, just as well think about it as acetylcholine for now. That would be just fine. And then you can apply it to other ones as well when you learn about different versions of these synapses. Okay, so at the axon terminal, if you recall, we did have that in order to get this neurotransmitter out, we typically have calcium that comes in, which is another ion. Calcium has 2 pluses on it. Ca double plus can come in and get that neurotransmitter to come out, right? And so if you look at the first picture that we started with, this one right here, right? We said that we're going to see sodium, we're going to see potassium, chloride. We've seen these 3, and now, as a reminder, we also have calcium here. So we've looked at all of the ions at this point. So calcium causes that neurotransmitter to start to get exocytosed from this synaptic end bulb. And exocytosis means it just comes out, and it starts to diffuse from one side of the synapse towards the other side of the synapse. And you can see here that over here there is a channel that we call a ligand-gated channel, which is going to receive that neurotransmitter, and then something is going to happen. And just as a reminder, looking at our first picture, right, what we want to talk about next is under channels is this ligand-gated channel right here. Okay, now ligand just means a chemical, right? And in this case, the chemical is the neurotransmitter. So basically, gated means that it's closed, right? Gated, closed, unless the ligand or the chemical, or in this case, the neurotransmitter, is present. And here we can see that it's closed, but that the neurotransmitter, the ligand, is going to start to diffuse and go towards it. And when it does bind to it, we're going to see that this gated channel does in fact open. Let's also remember that we do have our graph that we want to keep in mind, right? At any given spot, you're going to be at minus 70 millivolts, right? So that's our resting membrane potential. So keep that in mind as we move forward. And now what we're going to see is at this point in the membrane, this minus 70 at this point is going to change, right? You can still have minus 70 up here or over there, depending on what's going on, right? There's going to be other presynaptic neurons all over the place that are going to try to stimulate the postsynaptic neuron. But right now, this negative 70 millivolts, this resting membrane potential, just keep it in mind right here at this part of the neuron where we're having the conversation. So now what we want to do is we want to bring in that neurotransmitter. So keep this picture in mind. You see how that ligand-gated channel is closed. And now, the next picture, we're going to see how it goes from a closed position to an open position, right? So here it's closed. The ligand is not actually attached to the channel yet. But over here, right, we can see how this neurotransmitter starts to diffuse and eventually finds that ligand-gated channel right there. And you can see how those red dots are now attached to that blue channel, opening it up, right? Versus the closed position, right? Closed position, no red neurotransmitters attached. Open position, the red neurotransmitters attach and open it up. Okay, what is that going to do for us? Well, we want to remember that we have a lot of sodium that is found outside of the cell because of the sodium-potassium pump. So just like it's doing it here, it's also going to be doing it over here as well. All right, these sodium-potassium pumps are everywhere. So imagine a bunch of sodium outside the cell. And this creates a potential, right? And the potential is, in this case, the potential for the sodium to actually enter once that ligand-gated channel opens, which is exactly what's going to happen. Once it opens, this is a ligand-gated channel for sodium, and sodium will rush in. And it will rush in big quantities. A lot of sodium will rush in. Right? Once you open up that gate, sodium starts to rush in. So whereas the leaky channels saw a little bit of sodium leaking in, here we have a lot of sodium that just rushes in. What is that going to do to our resting membrane potential? Right? So don't forget, we have a resting membrane potential of minus 70 millivolts, right? But now all that positive sodium comes in, and remember this negative 70 is the inside of the cell, right? Because of this voltmeter, right? This sensor right here, that's what it's telling us, minus 70 on the inside. So now the inside becomes more positive, right? Because of sodium. So I'll draw it in purple, right? Well, this is purple. It's kind of a dark purple, but it's supposed to be the same color as this sodium right here. And this is causing our, let's not forget what we call it. We call it a depolarization. Okay, so we see this depolarization here because of, right? Because of sodium, and we sometimes use the word influx, right? Influx meaning inside, sodium going inside. And this is how that postsynaptic neuron gets stimulated. Now, if this signal is not too strong, right? Eventually, this depolarization will simply repolarize itself. So the signal won't go very far if, for example, it's just this one neuron that is doing it. Right? The signal will not go too far. Let's see how far this signal can go. Let's bring in some more channels. So, looking at our original picture, we have ligand-gated channels, which we just looked at, and then we have voltage-gated channels. We've already seen the leak channels. Let's look at the voltage-gated channels next. So, remember that we have this neurotransmitter in red binding to that ligand-gated channel. Sodium rushes in, that starts to depolarize the cell. And then let's add some voltage-gated channels to our picture. These are the voltage-gated channels next to the ligand-gated channel. And this one is a voltage-gated channel for sodium. Right? This one is for sodium. Whereas, and that one is in purple, and this one in pink is a voltage-gated channel for potassium. Let's not forget our ligand-gated channel is open. Our sodium ends up going in, and we will start to depolarize the neuron, right? So we start to depolarize the neuron. The next thing we want to do is realize what happens after this. And that's when we look at these voltage-gated channels. So whereas ligand-gated channels open with a chemical such as a neurotransmitter, voltage-gated channels open when you change the voltage. They are sensitive to voltage, and their shape changes when the voltage changes. So keep that in mind. In this case, the voltage is changing, right? We are depolarizing. So what's going to happen is that this one, right, this sodium voltage-gated channel is going to respond to this depolarization by opening. So the next picture would have that sodium channel opening up so that it would look like this, right? So we've got the sodium going into that ligand-gated channel, and notice what's going to happen next. The voltage-gated channel for sodium opens, and that's going to cause sodium to enter through the voltage-gated channel. That means you have even more sodium coming in, not just from the ligand-gated channel, right? Not just from the ligand-gated channel, but also now from the voltage-gated channel. And sodium starts to come into the cell. Well, what is that going to do to our graph? So we were at minus 70. Let's just pretend, right, that our ligand-gated channel brought us to, let's say, minus 65, okay, minus 65 or so. Our ligand-gated channel did that for us, but then the voltage-gated channel opened, and that started bringing us up even further to, let's say, minus 60, depolarizing us more and more and more. Well, next door to that sodium voltage-gated channel, you have a potassium voltage-gated channel. And this one is going to open when the cell becomes more positive. So as the cell becomes more positive, right, and you depolarize this way, eventually the sodium-gated channels will close, and the potassium channels, the potassium-gated channels, will open. Now, what is that going to do? Well, remember that our sodium-potassium pump put in a bunch of potassium on the inside. And if you remember your graph, you had that minus 70. You depolarized with the sodium, right? And then it became very positive because all that sodium came into the cell, right? Let's draw that in. All that sodium came in, but now you've got all this potassium that can come out. And when that potassium does come out because of that voltage-gated potassium channel, right, you end up repolarizing, right? So whereas the depolarization is due to sodium influx, the repolarization is due to potassium efflux, right? The repolarization. Okay, so this is one of the most important things to understand. Depolarization due to sodium influx, repolarization due to potassium efflux. Now, let's just assume that we just have this one presynaptic neuron stimulating this one postsynaptic neuron. Oftentimes, if it's only here, if the signal is only in this local area, it's a localized electrical event. and you get this depolarization that doesn't go too far. It doesn't go past negative 55, let's say, right? It stays closer to minus 60 or something like that, or minus 65. It doesn't keep going up too much. And therefore, the signal stays localized in this part of the neuron, right? If this happens, then we call this a graded potential, right? So if it's local and it just stays around here, okay, we call it a graded potential. Now, this doesn't mean that it will not cause a change in this neuron. The neuron will respond to it, but graded potentials do not cause the axon to fire the action potential. They stay localized within the dendrites, okay, within the cell body, and they don't move past that. They do not propagate down the axon. However, let's bring in another presynaptic neuron. Let's pretend that now not only do we have this neuron down here releasing its neurotransmitter, but you also want to imagine another presynaptic neuron releasing its neurotransmitter somewhere else. And you've got that ligand-gated channel here, so now you want to imagine that you have this sodium that starts to come in. From this ligand-gated channel, sodium starts to come in to that ligand-gated channel. This will be a more powerful event than in just one location, right? So then you draw your graph. You start at that minus 70, and you can imagine that, right, in this spot you would have that depolarization, but you would also have it in this area too. So both areas would start to depolarize, right? Both parts of this neuron would start to depolarize, and then you would have voltage-gated channels over here as well as over here, right? So you would have sodium that would rush into this sodium voltage-gated channel, but also you would have sodium that would rush in to this sodium voltage-gated channel. And you can see how this is a more powerful event, right? So before we had a graded potential only in one location, and now we can see that if we do this in several spots, we would get a more powerful event. And, right, this would be followed by a much bigger depolarization. Now, we should put that we have a value of minus 55 millivolts, which is known as threshold. Meaning that this minus 55, as long as you don't surpass it, right? As long as you do not surpass it, you end up with a graded potential. But if you hit that minus 55, this neuron feels that much bigger signal, and channels start to open much, much more, and ions start to flow in much, much greater numbers. And so you can imagine many more voltage-gated channels opening, and that would cause this depolarization to be much more powerful, reaching, right, not only zero, but actually going past it to plus 30. Because it goes past that zero. It goes so fast it can't really stop right there. So it goes to plus 30. And that's what's being depicted here. Right? And that's going to be followed, of course, right, by the potassium channels that are going to open on both ends while the sodium channels close. So minus 70, minus 55 is your threshold, right? And then what's going to happen is we depolarize first, right, all the way to plus 30, and that's from that sodium, but then potassium is next. So potassium, right, starts to come out. And that's going to cause our repolarization. And just like we couldn't stop at 0, right, we had a hard time stopping at 0, we went past it to plus 30, same thing is going to happen here. We cannot stop at at minus 70, because it's moving too fast, so we overshoot, and that is usually a minus 90 millivolts value, and then things tend to reset, right, towards that that minus 70 resting membrane potential. So this part right here, this section right here of the curve, if this is the depolarization, this is the repolarization, right? Depolarization, repolarization. This part of the curve where it overshoots and goes past that resting membrane potential of minus 70, right? We call this part the hyperpolarization. But then it does reset to the resting membrane potential eventually, and then it can do it again. So now we want to understand how we can get to that action potential. And let's not forget that the action potential moves into that axon and then starts to propagate, right? Whereas the graded potential becomes weak if it's not too strong and it stays within this area. The action potential does propagate. So keep this picture in mind, but I'm going to take away some of the clutter here, and we're going to use a cleaner picture for this. And we're going to simplify a little bit, and we're not going to keep opening and closing these channels. We'll just show the movement of ions for now. And for, imagine other presynaptic neurons all over the place having their effect. But for this picture, we're just going to have this one presynaptic neuron. And we're going to assume that the signal is strong enough in this position to move to an action potential that moves along the axon. So how is this signal going to propagate? Let's not forget we have sodium voltage-gated channels and potassium voltage-gated channels, and they're in sequence one after another. The idea would be that the sodium comes in and the potassium goes out. And if this is strong enough, then we'll start going into this initial segment of the axon, or sometimes called the axon hillock. And so what's going to happen in that axon hillock is that you would have another set of ligand-gated channels. And it would move sequentially, meaning like a domino effect, right? Like a domino effect, you would have the signal that moves towards the next section of these channels, where sodium comes in and potassium goes out. And always, at every single time you see this, you have to imagine that graph going from, right, the sodium brings it to plus 30 depolarization, the potassium hyperpolarizes it to minus 90, and then you get that repolarization at every single time you see these two channels next to each other. That's what you want to imagine, but then it happens again. And it's going to find the next sodium and potassium voltage gated channels. Once again, sodium goes in and potassium goes out. And this is going to keep happening, and so we can just draw arrows now in order to simplify the picture, right? An arrow going in for sodium and an arrow going out for potassium. Like a domino effect, it's going to keep moving down the axon, and this right here is called an action potential. Right? And the idea is it would move all the way down the axon until it gets to the axon terminals. Now, believe it or not, if we had to worry about every single piece of this membrane opening and closing, it would be slower if we could jump over big chunks of the membrane and only worry about the portions that we're jumping into. Let me try to explain this using this picture. So here I have drawn these cells that are insulating the axon. And these are known as Schwann cells. And they create these myelin sheaths, which insulate the axon, but also do something very important for the speed of the action potential. Let's put two pictures close to each other. So in one picture, we want to have the one that we just had, which is this one, with those gates one after another, right? And you can imagine this having to, you'd have to wait for these gates to do their opening and closing, then these gates, and so on and so forth. What if we put those Schwann cells on that axon? Well, now what happens is that we can actually jump to these sections that are found in between the Schwann cells, these sections here, known as the nodes of Ranvier, so that once the sodium and the potassium do their thing here, where the sodium goes in and the potassium goes out, what happens is the signal then can be felt further up ahead at these nodes. So you can jump over this whole big piece of axon and just go straight to that node. And at that node, you have sodium coming in and then potassium going out. And likewise, now you can skip this whole piece of myelin and go and jump to the next node, where you have sodium going in and potassium going out. This is much faster, and this is known as saltatory conduction. Now, the word saltatory just simply means jumping, because you're jumping from one node to the next. So instead of walking the axon, you're jumping over it. All right? So that you're going from this section of it straight to here and straight to there. This is much faster than the previous picture that we showed, which is known as continuous. Right? This one, where we have no myelin, is known as continuous conduction. And you see continuous conduction is common in small neurons. Why? because with small neurons, because they're already short, you're not going to lose much speed if you have continuous conduction. But in long neurons, you want to imagine a neuron of your sciatic nerve, for example, that has to go all the way from that sacral plexus, right? That sacral plexus where your sacrum is, right? All the way to your foot, let's say, right? Saltatory conduction is found in long neurons because this really helps with the speed, right? It allows for even a long neuron to be very, very fast with these extra myelin sheaths. So this is the end of this lecture. And of course, once we move down this axon, we have the axon terminals and we have the synaptic end bulbs. So let's not forget what happens there. We can look at this picture for that just to remind us, right? If we imagine this action potential moving this way, eventually, of course, it'll split into the axon terminals, go to the synaptic end bulb, and release a neurotransmitter with calcium, for example, right? And in that way, you can think about, right, this neuron that we thought of as a postsynaptic neuron, right? Once the signal moves down and eventually goes all the way to the synaptic end bulbs, right? It will become a presynaptic neuron for the next neuron in the circuit, right? And then you can imagine many neurons talking to themselves, communicating with each other, and firing action potentials to allow you to think, to allow you to move, to allow you to do all of your homeostatic functions, etc., etc.